Good morning, everyone. So we are getting slowly towards the end, but very exciting parts still have to happen. And I'm really thrilled, actually, to introduce Hannah Monyer. She comes from Romania. She studied medicine, psychiatry, neurology in Heidelberg, went to Stanford to do research in Stanford, and came back to Heidelberg, and now is leading a big group in Heidelberg. She is the queen of GABA, of inhibition. We will learn everything about GABA, so I'm really looking forward to her talk, but not only for the science, also because she, tries to get rid of dogmas. I think science is really hampered by dogmas, dogmatic thinking, and sort of like every so often we have to think outside the box. And I think Hannah is a prime example to do that. So I would like to give her the floor, and we should give her applause. The final thing I want to say is that there will no time for questions, unfortunately. So any questions that you may have, you will have to ask afterwards, after the talk. And I will not be present because I have to run also for my flight. So Hannah, take the floor.
Yes, thank you very much for the kind words of introduction and giving me the possibility to talk to you here. I mean, of course, I know that some of you are on the way out already, but I will tell you why, for those of you who are here, why working on inhibition is actually very exciting and why you should introduce it into your models. And my talk is called The Short and the Long of Inhibition because the GABAergic cells that I'm working on, most of them, they were thought in the cortex to be interneurons, and I will show you that there is quite a significant population that is projection neurons, and we think that they are very, very exciting. So the structure I will be talking about is the hippocampus and the hippocampal formation, and my lab works on spatial representation and episodic memory, concentrating on the GABAergic neurons in these structures, and more recently also in the cortex, and I hope I will convince you why these cells are the pacemakers of most rhythms in the brain and why they are important for any type of cognition that you're thinking of. So we do mouse genetics, we implant tetrodes into these structures, and we can resolve the single units and during behavior, but also during simple exploratory behavior. You probably all know the seminal work of O'Keefe and of the Mosers, who discovered the place cells in the hippocampus and the other spatially tuned cells, the grid cells in the medial entorhinal cortex. So for those of you, just very, very brief, the introduction, if a mouse runs in an environment as shown here, you see the trace in gray, and these are the action potentials of an individual neuron. This neuron in that particular location will fire there and only there. And if our tetroids do not move next day, this neuron will fire there. And O'Keefe proposed that it is the place cells in the hippocampus that built the cognitive map. In the medial entorhinal cortex, as you will see these pictures again and again, an individual neuron fires in multiple places. So this is the firing pattern of one single cell, and this cell fires in these locations. And if you look at it, it forms a grid pattern. And so the place cells tell you where you are in space. The grid cells are very important for computing the distance that an animal traversed, and also it does vector integration, and I will show that to you in just a second. Now, here again, we have the different structures, the hippocampus and the entorhinal cortex. It is currently thought that the medial entorhinal cortex, where the grid cells are, but also other spatially tuned cells, that they give to the hippocampus the information about space, and that the lateral entorhinal cortex provides the information about objects. So what happens where is given to the hippocampus, where this is considered to be united in a common representation of a particular episodic event. And if you think of your memories, most of your memories are set in a location, because from all sensory cortices, you have direct projections to the hippocampus. So again, in the medial entorhinal cortex, We have grid cells that measure distance. We have border cells. So these are heat maps, right? So you have individual neurons where they fire. Here, a border cell would just fire at the border, and this cognitive map is basically hung up then, right? I mean, that's the fixed point. Then we have head direction cells. These are cells that fire only when the animal walks in a particular direction. And in the lateral entorhinal cortex, we have object cells. So have a look at this. So this cell would fire just here when an object is in the left corner. This cell would just fire in that right corner. And then we have some other non-spatial cells. I do not have time to go into this. It's not that clear cut as I show it here, because actually we and others have provided evidence that you have some non-spatial coding in the medial entorhinal cortex and that you have some spatial cortex also in the lateral entorhinal cortex. But in a simplified version, one would say the MEC codes for space, the LEC codes for objects. You also have object vector cells. So these are cells that fire where an object had been. And then this information is directly transferred to the hippocampus where you would have this cognitive map. So my lab works on this, and we use these spatially tuned cells as readouts. We actually are not interested in these cells. These are all excitatory cells, but we are interested in those cells, the GABA cells, which coordinate these neuronal ensembles. And now I will show you why they are so interesting. So if I use a metaphor, I would normally say the excitatory neurons are the players in an orchestra, right? They play the different instruments, they play the music. But it is the GABA cells which are the conductors of this orchestra. So if you stay with one image, the GABA cells, they make the synchronous activity. And without that, I will show you the animal are seriously impaired in any type of cognitive memory. Now, already Cajal, actually, if you look at his wonderful drawings, this would be in the cortex, pyramidal cells in the different layers, which to me look fairly boring. They all look the same. And look at these beautiful GABA cells. They come in different flavors, and they do many different things in the system. We work mainly on the fast spiking cells, but of course, they do very different timing. As I will show you here, for instance, this would be some filled GABA cells in the hippocampus, and you can tell by their shape that they are very, very, very different. To make it very simple here, if we look at the pyramidal cells, these parvalbumin GABA cells, they are also the fast spiking cells. They inhibit excitatory cells perisomatically, so they would control the output of the cells. There are other GABA cells, like the somatostatin positive cells, and they target exclusively the dendritic part of the neurons. So they would actually control the input into the cells. So these different GABA cells, they share functions in the network and control the timing when excitatory cells are active. And then there are some other GABA cells that just talk to each other, and they are very interesting too, and I will tell you in a second why. So this is what my lab works on these directors of the orchestra, if you want. So, and all the way back, when I was a postdoc with Peter Seeburg, I cloned some of these, when I came back from Stanford, I cloned some of these glutamate receptors, right, that would excite the GABA cells. And by single cell PCR, I found out that interneurons have a different molecular makeup in the expression of their genes. They have different calcium channels, they have different potassium channels, they have different NMDA channels, so the kinetics of these NMDA receptors is different, and they also have different AMPA receptors, which are the major excitatory receptors, or that mediate excitation, so that would recruit these GABA cells. So their kinetics is faster, they're calcium permeable, and lots of other features. So the interneurons, the GABAergic interneurons, they have a different molecular makeup, and my lab used for many years mouse genetics to play around with these receptors. This was very strongly model-based. What would happen in a network if we, for instance, reduce excitation? And I will make it very short. If we do not change receptor expression in excitatory cells and only in interneurons, and to reduce their recruitment, the animals have serious problems in many behavioral tasks looking at memory, at spatial memory. So here already in the old days, we could show that if you look at the field potential as shown here in a normal hippocampus, if you look at the when interneurons fire, they are like a clock. They fire at a particular phase of this local field potential. And if we reduce the excitatory drive by taking out one of these receptors, so we just reduce the drive with which you recruit these receptors, we can still activate these interneurons, but they fire in and out of phase. And this temporal coding is very, very important for the network. It does not just matter that the neuron fires. It matters when it fires and with whom it fires to build a particular representation. And this is just a memory task. Here we test working memory. If we take out one of these AMPA receptors just in these fast spiking cells, and here the mouse just has to go to the right side, it explores the environment. If you open the formerly closed arm, a normal mouse is inquisitive, it is interested, it will remember where it had been before, and it will now go in the previously closed arm, given that it remembers where it had been 5 seconds before. And in these knockout mice, they're almost at chance level. And we really change just a little bit the excitatory drive onto these interneurons. Now, just for you to sort of think, the GABAergic cells are a minority of cells, so they are, depending on the brain regions, 10 to 20% of the number of the excitatory cells. So it suffices to do the small manipulation to reduce the drive on the fast-spiking GABA cells, and you almost have a phenotype as if you took out this receptor in the entire brain. Now, a subject that is very, very dear to my heart, and we still continue to work on that, you probably know that in the adult brain, basically almost everything communicates via chemical synapses. The neurons communicate via chemical synapses, but not interneurons. So interneurons have, in addition to chemical synapses, they are connected on their dendrites by gap junctions. And these gap junctions, again, based on network modeling, we could predict that if we take these gap junctions out, it will not affect the network completely, so you can still recruit the cells, but you know they are not as precise. Now again, these gap junctions are just on interneurons and just on interneurons of the same kind. So they make some local network that we think are very, very important. If you're interested in that, I can tell you more about this after the talk. So if we take these gap junctions out in this memory task that I showed you, the mice are at absolutely chance level. So again, we do nothing to the pyramidal cells, just to these interneurons. We take the gap junctions out, and the mice do not have spatial working memory. Now, this, if the animal explores an environment as shown here, in this, the gene is called connexin 36. So we take out this connexin 36, and then there are no gap junctions. There is no other gene connecting making gap junctions in the mouse or in the rat. So if we take that out in the interneurons, this would be some normal, beautiful place fields in the wild-type mouse. Above you see their maximal firing rate. So this neuron would fire here, and you don't have to be, a mouse specialist or working with such data. In the connexin 36 knockout mouse with no gap junctions, you can see that these place fields are smeared out, and they're also not stable. If we look at this mouse an hour later, this cell would fire in this location. If the tetrode has not moved, also next day it would fire in the same location, but not so in the connexin 36 knockout mouse. In a knockout mouse, these place fields are unstable, and that accounts for the impaired memory in these mice. Now, I will go to the medial entorhinal cortex, and I will show you that these GABA cells are very, very important to build grid cells. And this is, again, considering modeling data when the grid cells were first found. What was very peculiar was this grid-like pattern, and one tried to figure out how is the connectivity and what model would actually explain this peculiar firing pattern of the grid cells. Now, we and others actually found out that excitatory neurons, the grid cells, they do not communicate with each other. So the excitatory neurons in the entorhinal cortex, there is no connection between them. But each grid cell communicates with an interneuron. So the communication goes directly via the interneurons, and the models predicted that under such conditions, the interneurons should also have a grid-like pattern, and it should be either a grid-like pattern or an inverted grid-like pattern. So when these grid cells were found, it was really not clear what the connectivity was, and this is really work of the last 10 years where we and others figured out how the connectivity in the medial entorhinal cortex is. And again, the interneurons play a very, very important role because they connect, actually, they form the connection, the indirect connection between grid cells. And I will now show you how that looks in the mouse. So if we record from tetrodes, and we use the activity from neurons that are from the same tetrode, and you do a cross-correlogram, you can actually look what the putative monosynaptic connection is between cells recorded on the same tetrode. Now, what you see here on the left is a grid cell, so this neuron fires in these 3 places. And the interneurons, they fire all over the place. So they do not have much of spatial information. And what this cross-correlogram tells you that this is a putative monosynaptic connection, and we can then quantify the data and actually say that it is the grid cells that make the most connections onto the parvalbumin cells. And also the parvalbumin cells receive the most input from the grid cells. So this is the major connectivity. Grid cells project onto parv cells receive input from the grid cells. But if that is the case, and the grid cell have this hexagonal pattern, why do not interneurons have? How come that this pattern is not transferred onto them? And the next slide will tell you why. So we have in our recordings, I think we had about 1,100 neurons, 1,000 grid cells, and 100 fast spiking cells. We had some cases where the same interneuron, the same fast spiking parvalbumin cell, received input from 3 different grid cells. And what you can tell now is that these grid cells do not have the same phase, right? They fire in different locations in space. And that accounts for why the interneuron would fire all over the place. So multiple grid cells connect onto one parvalbumin cell, and therefore the parvalbumin cells, it's not just an individual grid cell, fires in multiple places. And this rule actually holds true for many other brain regions. In the visual cortex, for instance, you have orientation selectivity columns, and the fast spiking parvalbumin cells is exactly the same. They do not have a preferred orientation. They respond to lines of different orientation. In the olfactory bulb, it's exactly the same. The other neurons, they respond to particular odors, but not so the interneurons. The interneurons are input gain control neurons, and this is what we see on this particular time. You have to excuse me. So I'm getting into action here. So this is very exciting. So the interneurons are very, very exciting. So and this is what we figured out here. And now I will tell you something that we are very much interested at the time, because for years these recordings have been done in mice that explored a normal environment. But what our field does is to put mice now in a more complex environment, in different rooms with different colors, in rooms with objects. And what we now find is that interneurons actually do code for particular features. And what you see in this particular example done by Antonio Caputi in my lab, this would be in normal environment. You see this beautiful grid cell, here one and here one. But if you now put an object in the environment, you can actually tell that we find some grid cells that do not keep their firing places where they had fired before. But interestingly, the interneurons that before were not specific, they now fire in very, very close to the object, we actually think, and that is my current working hypothesis, that interneurons are very important in the selection of the network that will be active in a particular location. So that is a very big project that we pursue. So interneurons, they can become spatially tuned in particular environments, and they will lead to the selection of a given neuron. And also when you let the mouse run, in one and the other direction, here you would see an interneuron that fires just in one direction, but not when the mouse returns. So they are also directionally tuned, and this is something that we pursue. So interneurons do code for particular features, and they're probably involved in selecting the network in a given environment. So the work that I've presented to you here was done by Christina Büttferung and Kevin Allen in the lab, and the most recent work is done by Antonio Caputi and Xu in particular. Now, I come to something that is very, very dear. I still have 10 minutes. I come to something that is very, very dear to my heart, and I think that that is very important for young people, that many interesting findings in the lab, not only for us, but in general, when you do experimental science, is serendipitous findings. You need to actually be prepared to see something that you did not expect. So what I had told you before is that in a given network, here we would have a pyramidal cell, you have interneurons, and they are called interneurons because their axons do not project out of the local network. So most GABA cells in the cortex are interneurons, and it is to such an extent that the GABA cell was always called interneurons. You find it in the literature, GABAergic interneurons. So one day I come to the lab, and what Antonio showed me was that there are some GABA cells that are not interneurons, but that are projection neurons. And moreover, they project very far away in the cortex, but also across hemispheres. And in the target area, they inhibit selectively GABAergic interneurons. So you have inhibition of inhibition, and you get disinhibition, right? And we think that these projection neurons are the big coordinators if you want to coordinate activity between the frontal cortex and the visual cortex or across hemisphere. And that is another very, very important subject that we pursue currently at the moment. So this was the original finding. The channelrhodopsin and EGFP was injected such in a mouse, in a Cree mouse. That's not important. But what you see here in the medial entorhinal cortex, it is only GABA cells that light up. So in essence, we should have seen the axons only confined to this region. But no, what Antonio saw was in the hippocampus, so the target area, he saw these long axons. So what you then do, you can make slices, you can patch the cells. So we were interested, what is the target cell? And I have shown you that already. So this is a symmetric synapse. Only GABA cells make symmetric synapses at the EM. So if we optogenetically, I go back, I don't know how to go back. Yes, so what we can do, we make slices, we inject here, we wait for 3 weeks, we make slices of the hippocampus, and now we stimulate optogenetically these axons that express channelrhodopsin, and then with a patch pipette, we patch hundreds of neurons around, 20 neurons, not hundreds, but 20 neurons around on one day, and we identify which cells respond when you shine blue light onto the slice. And there, Sarah found that these target cells are exclusively interneurons. We can fill those cells, so this would be a response such as a current induced that you can block with gabazine, and that tells you this is a GABA response. It is an inhibitory response, as you can also tell by the reversal, which is the GABA reversal potential. You can fill the targeted cells in the hippocampus, and from the firing pattern, from the morphology, we can tell that these long-range GABA cells, they inhibit exclusively interneurons far away. And what I do not have time to show you, if we inject the virus into the hippocampus, this is a bidirectional connectivity. You find long-range axons in the medial entorhinal cortex, and they also target exclusively GABAergic interneurons. So we injected many brain areas. Let me show you here, for instance, just the motor cortex. If you inject and you just express with a virus this channelrhodopsin and the EGFP in the motor cortex, have a look how many areas are targeted. So in a particular region, you can have many regions that are downstream that are targeted. Currently, we work on the medial prefrontal cortex with this particular target area, which is involved in pain perception. And if we just stimulate the axons, we change the nociceptive response in the live animal by just affecting a few of these long-range GABA cells. So if you're interested, so this is work that we are submitting now. And so this would be, for instance, from the medial prefrontal cortex. All these areas are, for instance, involved in pain perception, also in the human, and they are connected bidirectionally by GABAergic projection neurons, and we currently work on this particular projection. I do not have time to go into this. Here would be the injection site, for instance, and in the target area, what is very, very interesting, the axons, they run in the first layer, so they run just in the superficial layer, and also they target only interneurons in layer 1, which then affect the columns. So we see the change response in layer 5 pyramidal cells, but that results from these interneurons that are exclusively in layer 1. So this is a current research topic. And this work that I have presented to you, the finding was made by Antonio Caputi and Sarah Meltzer in the lab, and they have been aided by these other people. And I now come to the very last point, which has translational potential that we pursue in the lab, and it is also about GABAergic projection neurons. And this is published work by Magdalena Schlesig in the lab, and I will make it. This is a neuron paper, and this is the cartoon thereof. So the hippocampal formation, the medial entorhinal cortex, the lateral entorhinal cortex are all controlled by a pacemaker nucleus, and that is the medial septum. The medial septum gives the rhythm, and that is when an animal explores, but also for us humans, the major rhythm in the hippocampus is the theta rhythm. So when I walk, that is what the EEG would actually show. And it is on this rhythm, which is the background on which the firing occurs, and particular cell types, they fire only at certain phases of the theta rhythm, right? So the theta rhythm is like a clock that gives this downstream structure, this basic activity. Now, why are we interested in that? We are interested in that because we actually think that in mild cognitive impairment and also in Alzheimer's diseases, this structure is affected first. And this is work that we are currently pursuing in the lab. But first, let me tell you that the medial septum has two types of GABA cells. One are these fast spiking parvalbumin cells, and what they do, they affect the theta rhythm. And you have another type of GABA cell, and these are the calbindin positive cells. They also project to all these downstream areas, and what they do, they increase burst firing, and they also affect phase precession. I do not have time to go into this. As I said, this is published work if you're interested and you work in this field. So these two GABA projections, they share the task and they do different things downstream and control these downstream areas. Now, why do we think, or why did I have the idea that they might be involved in neurodegenerative diseases? So I had introduced the grid cells before, and it was in my lab after the Mosers discovered that by doing particular genetic modifications, we were the first to show in the live animal that grid cells are important for path integration. Path integration is a phenomenon that has been known since the 60s in rats. If a rat walks in a room, let's say like this, in zigzag, the rat also in the dark can return in a straight line, and so do we. And for this, it was always, as soon as the grid cells were discovered, it was thought that it is grid cells that do this vector integration. And our lab has shown that. That's a paper by Hill et al. That we published in Nature Neuroscience a few years ago, that if you mess around with the grid cells, and they do not fire in this regular pattern, the animal cannot do path integration. Now, path integration is amongst the first symptoms that you have in mild cognitive impairment and early Alzheimer. And it is this and the episodic memory. So when we saw this very, very fast firing rate of the medial septum cells, we wondered whether they could be particularly vulnerable because they have these long axons. They need lots of LTP, not LTP, they need lots of metabolic, they have a high metabolic need. So we predicted that it is these axons that might be affected before the downstream networks show any impairment. And that was indeed the case. So I don't have time to show you, but the local synaptic activity, the spontaneous EPSPs, the spontaneous IPSPs are unchanged in the entorhinal cortices, but very early in different models of Alzheimer, these axons are affected. This is just to show that also in the human, path integration is calculated. We use exactly the same approach in our mice. You actually look this ability, of course, in the dark, because path integration, that's very, very important. I have to be able, and also the animal, to do this in the dark. It is not based on visual perception. It is just based on the input that you have from your own body, from your joints and from the vestibular system. And again, it is in Alzheimer and already in mild cognitive impairment, very, very early affected. And this is now what we are doing currently. So here you would have in the wild type mouse, if we inject the virus to label these projections, you see there the axonal terminals in the hippocampus, in the wild type, in the medial entorhinal cortex, or the lateral entorhinal cortex. The lateral entorhinal cortex is in fact the very, very first to be affected in Alzheimer, reason why patients also cannot smell well, because the lateral entorhinal cortex does that as well. And here are two models of Alzheimer mice. One is the 5XFAD one. This is a tau model. And you can see this very, you see it actually by eye. You don't even have to quantify that. And these are 5-month-old mice. These are not mice that are a year old, or when you read papers, normally, you see these mice models used at 1 year and a half or at 14 months. This is in 5-month-old mice where the local networks do not show any sign of synaptic impairment, but we do see that these long-range septal neurons are affected. And this work is done currently in the lab by Beate at home and Jinjai Pei, and they're helped by Elte Fuchs and Kevin Allen. So in summary, what I have shown you is that GABAergic local interneurons are very, very important for local networks, and affecting them in a small region like the hippocampus, you will have impaired spatial memory, impaired episodic memory. I showed you that for a long time it was believed that these cortical GABAergic cells are interneurons. We found that a subfraction of them are actually projection neurons, and they have a very, very strong effect in the downstream area because they inhibit selectively local GABAergic interneurons and have a great power over large circuits. And then finally, I have shown you that these GABAergic cells, the projection neurons, that our current idea is that they're very vulnerable in neurodegenerative diseases such as Alzheimer. I personally think it's not just Alzheimer. I think in Parkinson it will be the same. So wherever we have long-range projection neurons and there is some impairment, and actually I did not tell you yet, but we can trace mitochondria in vivo, and we can see in these axons fragmented mitochondria. And for a long time, Alzheimer always has been, or has been considered also a mitochondrial, it was even thought that mitochondria might be causally involved. So to make a long story short, what we pursue currently is exactly this trace that these cells, we do not say that they are causing Alzheimer, but we say when the system is impaired very, very slightly at the beginning, these cells are more vulnerable, and they give us the potential for a differential approach at early stages. Thank you very much for your attention.